Welcome to this video where I look at structure and function in proteins. Here we can see the general structure of an amino acid. Proteins are made up of amino acids which are bonded together through peptide bonds. Each amino acid has an amino group which is basic, which means that it can accept protons through the lone pair on nitrogen. It also has a carboxyl group which is acidic, which means that H+ ions can dissociate through breaking of this covalent bond. The R group in amino acids gives each amino acid distinct chemical properties. Different amino acids have different R groups. For example, if the R group is H, the amino acid will be glycine. If the R group is SH, the amino acid will be cysteine. Below, you can see two cysteine residues depicted by these straight lines. The R group of each cysteine, the SH, is shown. Oxidation will result in a disulfide bond forming, as shown here. And this now is called cysteine. Two cysteine residues bonded together through their R groups by forming this disulfide bond. As you will see later, the disulfide bond is very important in stabilizing protein structure. Two amino acids can form peptide bonds, and here the carboxyl group of one amino acid will form a peptide bond with the amino group of another amino acid. This will release water through a condensation reaction. Water is formed from the OH of the carboxyl group and the H of the amino group. Now you can see that the peptide bond formed is highlighted in red and water has been released. Remember from my previous videos the N-1 rule. If we count the number of amino acid in this peptide, so here we have one amino acid and another amino acid, we take away one and that will tell us the number of peptide bonds. Here we formed a dipeptide, which is two amino acids bonded together through a peptide bond. Here we look at secondary structure, which is the way that a polypeptide chain folds. Now here, the first secondary structure I'd like to look at is alpha helix. You can see a polypeptide chain, which is a long chain of amino acids which are bonded together through a peptide bond. It's stabilized by hydrogen bonding as shown. The alpha helix is a right-handed helix, which means if you look from the top, you will see that the helix moves in a clockwise direction as it moves away. The alpha helix has an average of 3.6 amino acids per turn. Alpha helices are very common in proteins. Another important secondary structure is the beta pleated sheet. And this can be found in structural proteins such as silk, which has very high tensile strength. The beta pleated sheet can be formed between different polypeptides or within a single polypeptide. So, for example, we can have one polypeptide here, another here. And you can see the hydrogen bonding, which stabilizes this secondary structure. Beta pleated sheet can have strands of polypeptide which are parallel or anti-parallel. Let's look at the parallel arrangement first. Let's say that we have polypeptide chains which move in the same direction. They are stabilized by hydrogen bonding. This is the parallel arrangement. In the anti-parallel arrangement, you will have sections of polypeptide which run in opposite directions to each other, and they interact through hydrogen bonding. Now, in the antiparallel arrangement, this is commonly found within a single polypeptide, like this. Now, here we can have a hairpin turn, and the hairpin turn basically connects two strands of beta pleated sheet together. Next level of organization in a protein is a tertiary structure, and this refers to the overall three-dimensional shape of the protein. So we have different levels of organization. The first level of organization is the primary structure, which is the amino acid sequence of the polypeptide chain. Now, the polypeptide chain can fold to give different secondary structures, and then they arrange themselves in space to form a specific tertiary structure. So as you can see, there's a level of organization, there's a hierarchy here. You start with the primary structure, then the secondary structure, and finally the tertiary structure. How is this tertiary structure stabilized? What determines the shape of the polypeptide? Well, there are different interactions between R groups of different amino acids. You can see here a disulfide bond between two cysteines. You can see here an ionic bond between charged R groups. Here we have hydrogen bonding between different R groups of different amino acids. Here the R groups are non-polar, giving rise to hydrophobic interactions. In summary then, the primary structure refers to the amino acid sequence of the polypeptide. The polypeptide can fold to give secondary structure. Examples are alpha helix and beta pleated sheet. The secondary structure is stabilized by hydrogen bonds. The tertiary structure is the overall 3D shape of the protein. The tertiary structure contains the primary structure and also different secondary structures. It's worth noting that the amino acid sequence of the polypeptide will determine which secondary structures form and will also determine the tertiary structure of the protein. Fourth level of organization is called the quaternary structure. And this means there are two or more polypeptide chains which are interacting with each other. An example is found in hemoglobin, which contains two alpha chains and two beta chains, and these are color-coded. Interactions between these four polypeptide chains can occur through hydrogen bonding and also through hydrophobic interactions. Hydrophobic interactions make up the bulk of the interactions within hemoglobin. Finally, we can have ionic interactions. Ionic interactions are found in the form of hemoglobin which is not bound to oxygen. Remember that we're looking here at interactions between different polypeptide chains. Another example of quaternary structure is found in antibodies. Each antibody has four polypeptide chains, two light chains shown as L and two heavy chains shown as H. Each chain is a single polypeptide. So we have four polypeptide chains, and they are stabilized firstly through disulfide bonds between cysteine residues within one polypeptide, as shown by the arrow. How are these polypeptide chains held together? Through disulfide bonds between different polypeptide chains, as shown here. Now this is a very, very stable structure because it's stabilized by covalent bonding, both within the polypeptide and also to form the quaternary structure. Let's now go beyond the spec to look at structural motifs found within proteins. An example of a structural motif is a beta barrel, as shown here. you can see that the beta barrel looks very much like a beta pleated sheet. It's an example of a beta sheet, and each arrow shows beta strands. Structural motifs are super secondary structures because they contain elements of secondary structure. In the beta barrel found here, you can see that it's crossing a plasma membrane, a lipid bilayer, and this kind of protein is called a porin. It provides a channel through which hydrophilic and ionic molecules can cross the lipid bilayer. To achieve this, you need to have R groups of amino acids on the outside to be hydrophobic, so they can interact with the hydrophobic tails of the lipid bilayer. Internally, the R groups pointing inwards need to be hydrophilic, so they can interact with the molecule that is to be transported. Examples of porins are, for example, LAM-B, found in bacteria, and this is involved in uptake of maltose. Another protein which contains the beta barrel is called Tol-C. This is also found in bacteria, and it's responsible for efflux of drugs, giving rise to drug resistance. Another important feature of a protein is the protein domain. What is a protein domain? Let's have a look at this example of something called HSP70, which is heat shock protein 70. What does HSP contain? It contains two domains, the ATPase binding domain, the substrate binding domain, and the lid. So the ATPase domain is a conserved amino acid sequence. So ATPase domains in different proteins will have a similar amino acid sequence. Because it has a similar amino acid sequence, it has a conserved tertiary structure. Each domain will have an independent function. In HSP70, the ATPase domain will bind to ATP and hydrolyze it, which is heat shock protein 70, is very important in the process of protein folding. It's what we refer to as a chaperone. What chaperones do is they aid in protein folding. Let's have a look at the ribosome here, which I've drawn. The nascent polypeptide chain is emerging, but it hasn't been completely synthesized. So, because it hasn't been completely synthesized, it cannot fold to give a specific tertiary structure. This increases the likelihood that polypeptides such as this will aggregate. They will bind to other polypeptides which are unfolded and just clump together. This can be very harmful. What HSP70 does, it comes around and it internalizes the nascent polypeptide. It prevents it from interacting with other polypeptides, and this prevents protein aggregation. When the full polypeptide has been synthesized, it is released by HSP70 and then it folds. The ATPase binding domain in HSP70 will bind to ATP and hydrolyze it. This will allow the internalization of this polypeptide. The substrate binding domain merely is the region of the protein which binds the polypeptide chain. The lid is a region of the protein which closes to internalize the polypeptide. Hope you found this video useful. Please also check out my other useful videos in biology on the website. Thank you.